Hello friends, welcome back to the Facts in Biology, a YouTube channel devoted to the aspirants of NEET examination who are continuously preparing their syllabus for upcoming NEET examination. Through this channel, I am helping them to prepare the different topics in detail, in depth. I myself, Anand Joshi. With my channel, I am trying to introduce you the variety of concept that has been required for NEET aspirants. If you find this channel useful, please subscribe, like and share to your friends also. Thank you. As far as our syllabus is concerned, we are studying the sexual reproduction in angiosperm, and in the last video you have studied the development of male gametophyte. In this particular video, we are going to introduce you to the development of female gametophyte right from the details. As far as the female reproductive whorl in an angiospermic flower is concerned, it is called as gynoecium, or more correctly it is named as pistil. Actually, the individual member of the gynoecium are named as carpel, and actually, as you know, they are megasporophyll, the modified reproductive leaves. A flower with many separate free carpel, we call it as an apocarpous flower, or the pistil is apocarpous, where each and every part of the pistil is free, carpel is free, and it is ununited. Most common example, you can have Michaelia champaka. We generally call it as in common language as sonsafa, where each and every carpel is free, and when they are free, they are said to be the apocarpous. Then the another condition is there where the carpels are fused together, and when the carpels are fused together, the flower is called as syncarpous. As there is one, or we can say many carpel, they are fused together. We commonly observe it in the plants like brinjal, or even tomato, or even the oranges. As far as a typical carpel is concerned, it consists of three components. Here you can see the first one, it is said to be the ovary, which is just above the thalamus, then an elongated stalk-like structure that is called a style, and at the tip of the style you can have stigma. The number of ovules which are present in the ovary, it varies greatly. In case of paddy, in case of wheat, or even in case of, we can say, maize, you can have a single ovule. And when single ovule is there, that ovary is called as uniovulate. And when you consider the other plants like tomato, ladyfinger, where more than one ovules are present, that condition is called as multi-ovulate or polyovulate. As far as the ovule is concerned, we are going to see the structure of anatropous ovule. Now each ovule which develop inside the ovary on a highly specialized nutritive tissue called as placenta, it is attached to the placenta with the help of a small stalk-like structure that is called as funicle. Here you can see the funicle which is attached to the ovule. On one side, on another side, it is attached to the nutritive tissue placenta. The place of attachment of funicle to the main body of that ovule is called as hilum. Now here you can see that this is the region where hilum is present. Now hilum is the place of attachment of funicle to the body of ovule. It is called as hilum. In angiosperm, most common type of ovule, it is called as anatropous ovule. Now in this anatropous ovule, micropyle is directed towards downside, or it is, we can say, parallel to the funicle, and it is present adjacent to the funicle. So here you can see the micropyle is there, funicle is there, they are parallel to one another, and when the micropyle is facing the downward side or it is parallel to the funicle, it is called as anatropous ovule. As far as angiosperms are concerned, you can find a different variety of ovules. Some of them we are going to see for NEET purpose. Now this is the first one. This is called as atropous ovule. Now where atropous ovule is concerned, you can see this is a micropyle and this is the chalaza. Now these are in a straight line, and when micropyle, chalaza and funicle, they are in straight line, it is called as atropous ovule. You can find the another type that is said to be the anatropous ovule that we are going to see in detail. Here the micropyle, chalaza and funicle, they are forming an acute angle, or micropyle and chalaza, they are in straight line. The condition is called as anatropous ovule, and most of the around 90% of the angiospermic plant, they contain anatropous ovule, and that's why it is called as a typical angiospermic ovule. Whereas amphitropous ovule are the ovule which are forming a angle of 90 degree between micropyle, chalaza and funicle. Micropyle, chalaza and funicle, when they form the angle of around 45 degrees Celsius, 45 degrees, that is called as amphitropous ovule. The fourth one is called as hemianatropous, where it is not exactly anatropous, where it is not exactly atropous, where the micropyle, chalaza and funicle, they are forming a right angle. They are at 90 degree, and that's why it is called as hemianatropous. Hemi means half, anatropous ovule we know, where the angle is around 90 degrees. Then campylotropous ovule, it is a curve. It is a horseshoe shaped type of ovule, and that's why it is called as campylotropous ovule, where you can see this is the horseshoe shaped structure, so it is campylotropous. The last one, it is called as sarcinotropous. Sarcina means spring, where the funicle is forming a spring like structure, that is called as sarcinotropous ovule. So atropous ovule, typically anatropous ovule, amphitropous ovule, hemianatropous ovule, campylotropous ovule and sarcinotropous ovule. These are the different type of ovules which are found in the angiosperms. But around 90% of the angiosperm, they will contain anatropous ovule. So typical anatropous ovule in angiosperm. If we see the typical anatropous ovule, a ovule consists of a central parenchymatous tissue. Now you can see this central parenchymatous tissue in the form of nucellus. In the center of the ovule, you can find this tissue. It is a parenchymatous collection of cells, and it is surrounded by two protective coverings here, number 1 and number 2. So these two protective coverings of nucellus, they are called as integuments. Now these integuments, when they are 2 in number, the ovule is called as bitegmic ovule. Many a time they are found 3 in number, that is called as tritegmic ovule, or in certain rare cases it is only 1 layer, and that is called as unitegmic ovule. But most commonly it is bitegmic, containing 2. The outer and inner integument, bitegmic ovule it will contain, and this entire structure is enclosed within the ovary, and with the help of placenta, funicle, it is attached to the ovary. A narrow opening which is present at the apex of ovule, we call it as a micropyle. Here you can see the narrow opening is present at the apex of the ovule, that is called as micropyle. Opposite to that, there is another group of cells that is called as chalazal cells, and this chalazal end, the chalaza, it is forming the base of the ovule, and it is exactly opposite to the micropylar region. Then within the nucellus, within the integument, the embryo sac is present within the nucellus. Very soon we are going to study the development of female gametophyte or embryo sac. It is present within the nucellus, and it is a multicellular structure embedded within the nucellus. Then if we see the process of megasporogenesis, how embryo sac develop, it is a process of formation of haploid megaspore from the diploid megaspore mother cell. Now this diploid megaspore mother cell become distinguished in the nucellus and more or less of the center, but towards the micropylar end of the ovule. Mind it well, this is the nucellar tissue, and out of this nucellar tissue only one cell will be selected. It is given nourishment, and then it will be differentiated to form megaspore mother cell. Now here you can see this megaspore mother cell is many times referred as archesporial cell. Now this archesporial cell, sooner or later it will develop into the megaspore mother cell. Now this megaspore mother cell, it undergo meiosis, and when meiosis takes place, we know there are formation of 4 cells. Now these 4 cells which are produced from meiosis of megaspore mother cell, they are 4 in number, and they are in a straight line, and that's why they are called as linear tetrad of haploid megaspore. So this is the inline, or we can say linear tetrad of megaspore. Now out of these 4 megaspore, in many plants, most of the plant, upper 3 micropylar, they undergo abortion, they are degenerated, they remain unfunctional, or in fact they are destroyed. Only lower basal chalazal, it become functional megaspore. Most of the angiosperm, they will have only one, and that is the lower basal chalazal megaspore become functional, and 3 micropylar megaspores are degenerated. Then this single functional megaspore, it acts as the first cell of female gametophyte. Generally, megaspore towards the center of functional megaspore, it is, we can say, when it develops, in fact it represents the first cell of the female gametophyte. It undergo 3 successive free nuclear division. And here you can see a single megaspore is developing into the 2, and then it is developing into the 4, and the division continues. At the end of the development, you can have 8 cells or 8 nuclei, out of which 4 are located at each pole. 4 towards the micropyle and 4 towards the chalazoline, and 1 nucleus from each pole. Mind it well, at micropylar end 4 nuclei are present, at chalazoline 4 nuclei are present, but out of these 4, 1 from each pole, it moves towards the center. 1 nucleus from each pole migrate towards the center, and they are called as polar nuclei. Now 3 nuclei towards the micropyle constitute the egg apparatus. Here you can see 3 nuclei, they develop into the cells, and they are representing the egg apparatus, and in the egg apparatus centrally placed egg is there, and it is supported by 2 synergids. Egg apparatus, it consists of large central haploid egg, and it is supported from both the side with the help of synergids. Now these synergids, they show highly specialized hair-like projections, and these hair-like projections are called as filiform apparatus. Now these filiform apparatus are very important during fertilization because they guide the pollen tube towards the egg. Whatever we can say pollen tube is going to enter into the ovule, it will be directed towards the female gamete, egg or ovum, with the help of these synergids. Once that is done, then the 3 cells which are present towards the opposite end, that is said to be the chalaza, they will have 3 cells again, and these 3 cells are called as antipodals. Now these antipodal cells, they are playing major role whenever there is a problem with the egg apparatus. If one egg apparatus is failed, then automatically this particular antipodal cells, they will be distinguished into the new egg apparatus. So double chances that if one egg apparatus fails, the place can be taken by antipodals. The 2 haploid polar nuclei of large central cell, they fuse to form diploid secondary nucleus or definitive nucleus just prior to fertilization, and after fertilization it will give rise to the triploid primary endosperm nucleus. Now at the end of development of female gametophyte, 7-celled 8 nucleated structure is formed, and that is called as female gametophyte, or that is called as embryo sac. Now this 7-celled 8 nucleated female gametophyte is the characteristic feature of angiosperm. This method of development of embryo sac from a single megaspore is called as monosporic. As single functional megaspore develop into the female gametophyte, it is called as monosporic. In angiosperm, the development of female gametophyte is endosporous. It occurs within the ovule, it occurs within the single structure of ovule, so it is called as endosporic. And female gametophyte, if you see this particular photograph, it is colorless, it is endosporic, and it is concealed within the ovule, or ovule are enclosed within the ovaries. Now as far as the development of female gametophyte is concerned in angiosperm, there are 2 major types of female gametophyte development patterns. The first one is called as polygonum type, where you can see a single functional megaspore is used. 3 of them are degenerated. Here these are the degenerated one, and a single functional megaspore develop into the embryo sac. So embryo sac or female gametophyte is the product of single megaspore, single functional megaspore. That type is generally found in the plants, and it is called as polygonum type of development of female gametophyte, as it is first studied in the polygonum plant. The second type is also there, it is called as lilium. Now this lilium, it was first studied in Fritillaria plant, where all the 4 megaspore are used, functional, and all the 4 megaspore will give rise to the female gametophyte. This is called as Fritillaria type or lilium type of female gametophyte development. As far as this particular video is concerned, I am very thankful to CBSE NCERT for providing the base of the syllabus, as well as the State Board of Maharashtra for providing the necessary information, and most importantly Mahatma Gandhi Vidya Mandir of Malegaon, Nasik. I am very thankful to them for supporting me in production of this YouTube channel. I myself, Anand Joshi, a teacher in biology. If you find this syllabus, if you find these videos very helpful for your preparation, subscribe, like and share my videos. Thank you very much.